Coming to our next chapter, acid-base balance. So, we have to maintain the exact appropriate concentration of H plus ions in the cells so that it won't affect the cellular metabolism. So the normal pH of the blood plasma is 7.35 to 7.45. So you have to maintain the pH within this range, that is between 7.35 to 7.45. Right? So before going to details of acid-base balance, first you should know what are acids, what are bases. So acids are substances which can donate the protons, and the bases are the substances which accept the protons. So that is simple. Acids are the substances which donate the protons and which accept the protons are bases. And what is strong acid, what is weak acid? So when an acid dissociates completely, we call it as strong acid, and when it dissociates incompletely, partially, we call it as weak acid. So that is strong acids and weak acid. So the pH at which it is off ionized is called as the pK value. Now coming to, so our aim is we have to maintain the pH range, that is plasma pH within the range of 7.35 to 7.45. Right? So in order to maintain that pH within the normal range, we require some mechanisms. So those mechanisms can be classified into three types, that is first line of defense, second line of defense and third line of defense. So we have three defensive systems which are responsible for maintaining the blood pH within the normal range. So what is first line of defense? First line of defense is nothing but blood buffers. So buffers are the first line of defense. So they are very quick. Quick in action, but they cannot go to completion. Right? So they cannot clear all the acids from the blood, but they are released into blood. They are quick to action. When you see, there are different types of buffers in blood. That is bicarbonate buffer, phosphate buffer, protein buffer, right? So these three are the buffer systems in the blood, and these three together we call it as first line of defense. So immediately they are released they come into action, right? Whenever there is a disturbance in the acid-base balance, that is whenever disturbance in the pH range, pH fluctuations are there, then the first things which come into action is buffers. So buffers, what are buffers? So buffers are the solutions which resist changes in pH when acid or base is added. So when you add acid or base, so these solutions they resist the changes in the pH. Such type of solutions we call them as buffers. So we have three types of buffers: bicarbonate buffer, phosphate buffer and protein buffers involved in the first line of defense. Right? So what is bicarbonate buffer? So this bicarbonate buffer has two components. One is sodium bicarbonate and other one is carbonic acid. Sodium bicarbonate and carbonic acid. So these are the components of bicarbonate buffer. So what is significance of bicarbonate buffer? Bicarbonate buffer is the most predominant extracellular buffer, right? And most of the buffering action is because of this bicarbonate buffer. And when you apply that Anderson-Hasselbalch equation to this bicarbonate buffer system, there we will get 7.4. So the pK value is 6.1 and the concentration of bicarbonate is 24 millimoles per liter, and that of carbonic acid is 1.2 millimoles per liter. So when you apply these in the Anderson-Hasselbalch equation, you will get value 7.4. Means when you are maintaining a ratio of 20:1, that is concentration of bicarbonate to that of carbonic acid should be 20:1. So when that concentration is maintained, then your blood pH will be in the range of 7.4. So that is the significance of bicarbonate concentration. So the concentration of bicarbonate is more than the carbonic acid. So this excess bicarbonate in the blood is called as alkali reserve. So we have to maintain so that much amount of bicarbonate, that is, it should be the ratio of 20:1. So because of that excess ratio, excess concentration of carbonic acid is required to maintain the blood pH in the range of 7.4. So that alkali is called as alkali reserve. Right? Coming to phosphate buffer. So we have seen that bicarbonate buffer is the major extracellular buffer. Phosphate buffer is the intracellular buffer. So the components of phosphate buffer are sodium, disodium hydrogen phosphate and sodium dihydrogen phosphates. These are the acid phosphate and basic phosphate and acid phosphate. So these are the components of phosphate buffer, and the concentration of phosphate buffer, when you apply these values in the Anderson-Hasselbalch equation, the ratio is 4:1. Right? And here the phosphate buffer is directly linked with the renal system, kidneys. Right? That's about the intracellular buffer, that is phosphate buffer. Coming to another buffer, protein buffer. So we discussed about bicarbonate buffer, phosphate buffer, and the third one is the protein buffer. These three constitute the first line of defense. Coming to protein buffers, there are some important proteins in the blood which show the buffering action. That buffering action of the proteins is because of presence of a particular amino acid called as histidine. So histidine has a imidazole group that has a pK value near to that of pH, normal pH. So because of that, the proteins which have histidine group, histidine amino acid, those show maximum buffering capacity. So albumin and hemoglobin, they have the histidine residues, and that is responsible for the buffering nature of this albumin and hemoglobin. Right? So major plasma protein is albumin, so it has the buffering action, and also hemoglobin has buffering action. So in acidic medium, what happens to proteins? So in acidic mediums, proteins, they exist in the positive ion. So amino group is converted to ammonium ion, NH3 plus ion, and in alkaline medium, they exist in the acidic forms. Right? So that's how they carry negative charge. So in acidic medium, they have positive charge. In alkaline medium, they have a negative charge. Right? Coming to how this hemoglobin acts as a buffer. Right? So hemoglobin has buffering action because of the presence of histidine residues. So there are 38 histidine residues in each hemoglobin molecule. So that is responsible for the buffering action of the hemoglobin. So that hemoglobin is involved in the transport of carbon dioxide from the tissues to the lungs with a minimum change in the pH of blood. So this transport is called as isohydric transport. So without affecting the pH, it transports the carbon dioxide from the tissues to the lungs. Right? So in addition to that, while it is transporting the carbon dioxide, it also responsible for the conservation of the bicarbonate. Right? So alkali reserve is maintained by hemoglobin. So these two are the important functions of hemoglobin in maintaining the acid-base balance. Now coming to how this hemoglobin is involved in this, how it acts as a buffer. Right? So as I told you, it carries the transport the carbon dioxide from tissues to the lungs. So in tissues, carbon dioxide reacts with water by the action of enzyme called as carbonic anhydrase. It is carbonic anhydrase and is converted into carbonic acid. So that carbonic acid dissociates into H plus ion and bicarbonate. So that bicarbonate is reabsorbed. Right? So that bicarbonate enters into the plasma in exchange for the chloride ions. Now what happens to that H plus ions? Those H plus ions, they react with the hemoglobin and that is converted into HHB. So that HHB is transported to the lungs. So in the lungs, that HHB reacts with oxygen and it releases the H plus ions there. Right? So that HHB with the release, when it releases H plus ions and accepts the oxygen, it is converted to oxygenated hemoglobin, right? Oxyhemoglobin. And what happens to those H plus ions which are released in the lungs? Those H plus ions in the lungs, they are they react again with the bicarbonate and is converted to carbonic acid H2CO3. That carbonic acid is again dissociated into carbon dioxide and water, right? So that carbon dioxide is expelled to the lungs. In expiration it is lost. So this is how the carbon dioxide which is formed in the tissues is carried to the lungs with a minimum change in the pH, right? So this is how hemoglobin is responsible for the hemoglobin acts as a buffer and responsible for maintaining the acid-base balance. So this is the significance of buffers, the first line of defense in the acid-base balance. Coming to role of respiration. So we have discussed about the first line of defense, that is buffers. The second line of defense is respiration. So respiration is also responsible for the regulation of acid-base balance. So what is the role of respiration in this regulation of acid-base balance? So whenever there is increase in the PCO2, partial pressure of carbon dioxide, then what happens? Then body tries to eliminate that excess carbon dioxide, so that it leads to increased rate of respiration. So respiratory centers are stimulated, so that leads to increased respiratory rate, hyperventilation occurs, and it leads to the removal of carbon dioxide from the body, right? So elimination of carbon dioxide occurs by hyperventilation. So whenever there is decreased PCO2 level, partial pressure of carbon dioxide is there, then what happens? Then the rate of respiration decreases. It leads to hypoventilation. So it tries to conserve the carbonic acid in the blood. Right? So in that condition, it leads to hypoventilation. Right? So respiratory mechanism is mainly responsible for maintaining the bicarbonate to carbonic acid concentration. It always tries to maintain the ratio within the range of 20:1, right? So that is the significance of respiration. So whenever carbon dioxide is more, that leads to hyperventilation. Whenever carbon dioxide is less, that leads to hypoventilation. So this is the second line of defense. Right. So in this picture you can see how that occurs. Whenever there is increased PuCO2, so that stimulates the respiratory centers, the respiratory rate is increased, and whenever there is increased respiration, hyperventilation, carbon dioxide is eliminated, and it leads to decreased carbonic acid levels, so that pH is again maintained within the normal range. Right. So whenever there is alkalosis, means whenever there is decreased PCO2, that leads to hypoventilation, and it tries to conserve the carbonic acid. It won't eliminate the carbon dioxide, but it tries to conserve the carbonic acid, retain the carbonic acid, so that pH will be adjusted. So depending on the PCO2 levels, either it may be hyperventilation or hypoventilation. Right? So by these two mechanisms, respiratory mechanism of acid-base regulation occurs. So by hyperventilation or hypoventilation. Coming to third line of defense. We have seen first line of defense buffer, second line of defense is respiratory regulation, and the third line of defense and the most effective type of regulation is renal regulation. Right? So renal regulation involves different mechanisms. So that renal regulation has 4 mechanisms by which it maintains the acid-base balance. Those are excretion of H+ ions, reabsorption of bicarbonate, excretion of titratable acid, and excretion of ammonium ions. So these are the 4 mechanisms by which renal mechanism or renal regulation of pH occurs. So when you see kidneys, the pH of the blood is 7.4 and the pH of urine is 6, right? So there is difference in the pH of blood and pH of urine. So from 7.4 to it is decreased to 6. So this is called as acidification of urine. So what happens in first mechanism, excretion of H+ ions, and the second one, reabsorption of bicarbonate. So in excretion of H plus ions are excreted. So when you see in the proximal tubules, PCT cell, in the PCT cell, proximal tubular cell, what happens there? There carbon dioxide and water react in the presence of enzyme called as carbonic anhydride, and that is converted to carbonic acid. Right? That carbonic acid is dissociated into H plus ion and HCO3 minus bicarbonate. That bicarbonate is reabsorbed, enters into the blood, so that is preserved. So what happens to H plus ions? That H plus ions, they enter into the lumen, tubular lumen, in exchange for the sodium ions. The source of sodium ions here, it is sodium bicarbonate. Right? So this is excretion of H plus ions. So there is reabsorption of generation of bicarbonate also. So what happens in this generation of bicarbonate? In case of generation of bicarbonate is, so that sodium bicarbonate is responsible for the is a source for the sodium ions. Sodium ions, they enter into the PCT cell in exchange for the H plus ions which are excreted into the lumen. Right? So after donating that sodium ions, which are sodium ions is released from the sodium bicarbonate, what happens to that carbonic acid? So we have to conserve that carbonic acid, so that is reabsorbed. Carbonic acid is, bicarbonate is reabsorbed, sorry. So bicarbonate is reabsorbed. So that mechanism we call it as reabsorption of bicarbonate. How that occurs? So that bicarbonate reacts with H plus ions and it is converted into H2CO3. So that H2CO3 dissociates into carbon dioxide and water, and those enter into again into the PCT cell. And in the PCT cell, by the action of enzyme carbonic anhydrase, it is again converted into carbonic acid, and that carbonic acid again dissociates into H plus ions and bicarbonate. Right? So bicarbonate, which is released from the sodium bicarbonate, right, is again reabsorbed. So this is the reabsorption of bicarbonate. So excretion of H plus ions and generation of the bicarbonate. Coming to the third mechanism, excretion of titratable acid. So in the DCT cell, distal convoluted tubule cell, what happens? Here, same carbon dioxide and water react to form carbonic acid by the action of enzyme carbonic anhydrase, and that carbonic acid dissociates into H plus ion and HCO3 minus bicarbonate. So H plus ions, they are excreted in the tubular lumen, and that H plus ions, they are buffered by the phosphate buffer. So phosphate buffer has two components, disodium hydrogen phosphate and sodium dihydrogen phosphate. So that disodium hydrogen phosphate donates the sodium ions. So in exchange for sodium, H plus ions enters into the tubular lumen. Right? Now H plus ions, they react with the NEH. They react with the NHPO3, NHPO4, that is, which is released when disodium hydrogen phosphate is converted to Na plus and NaHPO4, right? So that it forms sodium dihydrogen phosphate. So disodium hydrogen phosphate releases Na plus ions, and when it is released, the sodium hydrogen phosphate reacts with H plus ions and is converted to sodium dihydrogen phosphate, that is a acid phosphate. Right? So this is the reason why the pH of urine is different from the pH of blood. So H plus ions are continuously excreted into the lumen, so that pH of urine is decreased. Right? So that's why it is in the acidic range. Right? So those H plus ions which react with sodium hydrogen phosphate now is converted to sodium dihydrogen phosphate, is the acid phosphate, and that is excreted. So this mechanism is called as excretion of titratable acid. So what is titratable acid? So ml of n by 10 KOH required to titrate this sodium hydrogen phosphate to a pH of 7.4 is called as titratable acid. So you are taking that sodium hydrogen phosphate and you are titrating with n by 10 normal KOH to the pH 7.4. So how much ml is required, that is called as a titratable acid. Right. Coming to excretion of ammonium ions. So this also occurs in the distal convoluted tubule cells, DCT cells. Here, so here you can see the mechanism. So there is one enzyme called as glutaminase which acts on glutamine and converts glutamine into glutamic acid and with the release of ammonia. So that ammonia which is released in the DCT cell now enters into the tubular lumen. In the tubular lumen, it reacts with the H plus ions, it combines with H plus ions, is converted into ammonium ions. So H plus ions which are released into the tubular lumen, they react with the ammonia and they are converted into ammonium ions. So here the significance of this thing is it won't, so if H plus ions are excreted as such, it leads to further fall in pH below 4. So in order to prevent that, so ammonium comes into action, it reacts H plus ions, they react with the ammonium ions and ammonia ions and is converted to ammonium ions. So when it is converted to ammonium ion, it won't change the pH and it won't be below the pH 4, right? So that is the significance of ammonium ions, right? So how that H plus ions are generated in the DCT cell by the same action, by the action of same enzyme carbonic anhydrase, catalyze the reaction of carbon dioxide and water to the carbonic acid, and that gets dissociated into the H plus ions and bicarbonate. H plus ions they enter into the tubular lumen in exchange for the sodium, and the source of sodium is disodium hydrogen phosphate, right? And H plus ions they are buffered by the ammonia which is released by the action of enzyme glutaminase which acts on glutamine and converts that into glutamic acid and ammonia, right? So this is how, so H plus ions are buffered and that bicarbonate which is generated is again enters into the plasma, so that maintains the alkali reserve, right? So these are the 4 mechanisms by which renal regulation of blood pH occurs. So those are excretion of H plus ions, reabsorption of bicarbonate, excretion of titratable acid and excretion of ammonium ions. So this is the most effective type of regulation of blood pH. So when you compare with that of first line of defense and second line of defense, so which cannot go to completion, but here renal regulation is most effective. So it can bring the pH to the normal range by 3 to 4 days. So it goes to completion when compared to the first line of defense and second line of defense. Right. So coming to the acid-base disorders, we have discussed that the normal blood pH is 7.35 to 7.45. So whenever the pH is less than 7.35, that condition we call it as acidosis. So whenever the pH is more than 7.45, we call it as alkalosis. Right? So whenever there is decrease or fall in pH, we call it as acidosis, that is below 7.35. So when it is less than 7.2, it leads to depression of the central nervous system and it may lead to coma. So when it is less than 7.07, it leads to death. And coming to the 7.45 range, so when it is above 7.45, it is called as alkalosis, and when it is more than 7.6, it leads to death because of neuromuscular hyperexcitability, so that and tetany occurs. So those are the conditions which are responsible for the death of the cases in case of alkalosis. So acidosis and alkalosis, this depends upon the pH. Right? Coming to acidosis, we have 2 types of acidosis: metabolic acidosis and respiratory acidosis. Same way, we have alkalosis, we have 2 types of alkalosis: metabolic alkalosis and respiratory alkalosis. So what is acidosis? So we know that acidosis is when pH is less than 7.35, we call it as acidosis. Coming to metabolic acidosis and respiratory acidosis, first we will discuss about the metabolic acidosis. So metabolic acidosis is primarily, it is because of deficiency of bicarbonate. So the main reason behind metabolic acidosis is deficiency of bicarbonate. So whenever bicarbonate is deficient, that is we are not maintaining the sufficient concentration of alkali reserve is not maintained. So that condition leads to metabolic acidosis. So what is the reason behind this metabolic acidosis? Why there is decreased alkali concentration? Why there is deficiency of alkali bicarbonate? Here the reason is due to accumulation of acids, fixed acids. We have two types of acids, volatile acids and fixed acids. So there is increased concentration of these fixed acids, mainly lactic acid, lactate, pyruvate. So all those acids, they accumulate, and their accumulation leads to utilization of that alkali reserve. So that alkali reserve is used for the buffering those excess acids. Whenever that alkali reserve is used in excess, that leads to decreased bicarbonate concentration. So whenever the concentration is decreased, that is whenever the ratio is low, whenever it is less than 20:1, that condition leads to metabolic acidosis. Here the main reason is due to decreased bicarbonate concentration, and that decreased bicarbonate concentration is because of increased acids. So what are the causes here? So we discussed about the exogenous. So there is endogenous excess production of ions, acid ions. Acids are produced, lactic acid for example. So whenever there is excess lactic acid, lactic acidosis we call that. Whenever there is excess ketone bodies, we call it as ketoacidosis. So these are acidic compounds, so that affects the pH of the blood. They decrease the pH of the blood. So accumulation of these compounds in the blood leads to utilization of excess utilization of bicarbonate. Right? So that is the reason behind the metabolic acidosis. So when you take excess amount of acids, that also leads to consumption of the alkali reserve. That is one more reason. And renal insufficiency, when the renal system fails to excrete H plus ions and reabsorb the bicarbonate, then also it affects the bicarbonate to carbonic acid ratio, so that will be lowered, and in that condition also it leads to metabolic acidosis. And abnormal loss of bicarbonate also results in. So whenever there is failure to reabsorb, there is increased loss of bicarbonate. So whenever there is increased loss of bicarbonate, that affects the concentration, that affects the ratio of bicarbonate to that of carbonic acid. So these are the causes for the metabolic acidosis. Respiratory acidosis. So we have seen metabolic acidosis because of primary deficiency of bicarbonate, and that is resulted because of the excess amount of metabolic acids. Right? Coming to respiratory acidosis, here the main reason in case of respiratory acidosis is because of excess amount of carbonic acid. Right? So there is excess amount of carbonic acid which is responsible for this type of acidosis. So this carbonic acid and the regulation of respiratory systems, so it is a respiratory component. So whenever the carbonic acid is more, it leads to acidosis. We call it as respiratory acidosis. So the primary defect in respiratory acidosis is excess of carbonic acid. So why there is excess of carbonic acid? Why it is increased? So we can discuss that under two headings. That is, so whenever there is excess intake of retention of carbon dioxide in the body, that leads to excess amount of carbonic acid, and whenever there is difficulty in the elimination of carbon dioxide. So whenever we are living in an atmosphere where there is excess amount of carbon dioxide, and the other reason is whenever there is difficulty in the elimination of carbon dioxide from the body, and these two conditions will lead to increased amount of carbonic acid in the blood. So whenever carbonic acid is more in the blood, pH of blood falls. So that condition is called as respiratory acidosis. So what causes this respiratory acidosis? Right? So there are some important reasons. Those are depression of the nervous system. Right? So whenever nervous system is depressed, it leads to hypoventilation, and there is decreased elimination of carbon dioxide from the body. So whenever there is decreased elimination, it leads to excess amount of carbonic acid in the blood, and that leads to fall in the blood pH. So that is one reason. And other one is conditions whenever there is difficulty in the diffusion, impairment in the diffusion of carbon dioxide across the alveolar membrane. Right? So whenever there is impairment in the diffusion of carbon dioxide across the alveolar membrane, that also leads to excess carbonic acid in the blood. Right, that is in case of emphysema. So in that condition, clinical condition, there is increased carbonic acid, carbon dioxide, increased PCO2 levels, and increased carbon dioxide, increased carbonic acid, whatever it may be, right? And another condition is asthma, right? What happens in asthma? In asthma, you can see carbon dioxide cannot escape from the alveoli, so that leads to increased carbon dioxide concentration, carbonic acid concentration will be more in the blood. Coming to whenever there is impairment in the blood flow to the lungs, pulmonary blood flow is impaired, insufficient. In those conditions also it leads to excess amount of carbonic acid in the blood. So that is in case of certain conditions like congenital heart diseases. So these are the main reasons which are responsible for the increasing the carbonic acid level in the blood. So we have seen about the acidosis. There are two types of acidosis: metabolic acidosis and respiratory acidosis. Coming to alkalosis, we have two types of again alkalosis. Those are metabolic alkalosis and respiratory alkalosis. So what is metabolic alkalosis? So metabolic alkalosis is the primary defect in metabolic alkalosis is deficiency of carbonic acid. So why there is deficiency of carbonic acid? So the acid component is low and the base component is more. Here there is excess carbonic acid. The ratio of bicarbonate to carbonic acid is increased. So there is more bicarbonate than the carbonic acid. Right? So what is the causes for this carbonic acid excess and the deficiency of carbonic acid deficiency and excess of bicarbonate? Here there is deficiency of carbonic acid because of there is excess loss of HCl, right? So pyloric spasm leads to excess loss of HCl. In those conditions, pyloric obstruction and spasm, right? So whenever there is excess loss of HCl, so there is loss of chloride ions. So in order to maintain the charge, so sodium and potassium are retained in the body. So sodium, potassium, when they are retained in the body, it forms a salt called as sodium bicarbonate. Instead of sodium chloride, there is excess formation of sodium bicarbonate. Why this sodium bicarbonate is formed? Because there is loss of chloride. Why chloride is lost? Because there is loss of HCl. So whenever there is loss of HCl, chloride is lost. In order to maintain that salt, that NaCl, neutral salt, is replaced by an alkaline salt called sodium bicarbonate. So in this condition, there is loss of acid and there is addition of base. And the other condition is whenever there is excessive vomiting, that also leads to loss of acid. And the other reason is hypokalemia. So this hypokalemia is associated with metabolic alkalosis. So in all these conditions, the ratio is increased. So what is that ratio? Ratio is bicarbonate to the carbonic acid. It is more than 20. So alkali reserve is increased in case of metabolic alkalosis. So with the deficiency of carbonic acid. So that is the main reason in case of metabolic alkalosis. Now coming to respiratory alkalosis. So the metabolic component is bicarbonate and the respiratory component is carbonic acid. In case of respiratory alkalosis, there is deficiency of carbonic acid with no change in the concentration of bicarbonate. So there is no change in the concentration of bicarbonate, but there is deficiency of carbonic acid. So why this occurs? So this occurs in some conditions like, so respiratory, whenever there is respiratory center is stimulated by the some conditions like meningitis. So whenever there is meningitis, that stimulates the respiratory center. So there is increased increased elimination of carbonic acid from the body. So whenever there is increased elimination of carbonic acid, that leads to respiratory alkalosis. So there is excess loss of bicarbonic acid. So that acid is lost. That is a respiratory component, and that leads to a type of alkalosis called as respiratory alkalosis. And the other reason in case of respiratory alkalosis is and whenever there is stimulation of respiratory center by some compounds like salicylates, so salicylate poisoning also leads to respiratory alkalosis. And the most common condition is hysteria, hysterical conditions. Hysterical attacks cause hyperventilation. So there is uncontrolled emotions. Those uncontrolled emotions are nothing but hysteria. So that hysteria leads to hyperventilation, and hyperventilation eliminates more amount of carbon dioxide, and that leads to again metabolic, that leads to again respiratory alkalosis. So these are the reasons behind respiratory alkalosis. So what is anion gap? So actually we'll measure some cations and anions. So the unmeasured anions is called as anion gap. So always there is difference in the measured cations and measured anions. So that unmeasured anions is called as anion gap. So and the normal value of anion gap is 15 milliequivalents per liter, and the range is 8 to 18 milliequivalents per liter. So there should be in this range, and the average is 15 milliequivalents per liter. So when it is there in this range, then we call it as normal anion gap. So whenever it is decreased, we call it as high anion gap. Whenever it is increased, we call it as high anion gap. When it is decreased, we call it as low anion gap acidosis. So generally in case of acidosis conditions, this anion gap is affected, and metabolic acidosis, there is increase in anion gap, we call it as high anion gap acidosis. And in case of that is in case of diabetic ketoacidosis, lactic acidosis. And in case of diarrhea, there is normal anion gap, right? Though that is the significance of anion gap. So that is altered in case of metabolic acidosis. Thank you.